Hello everyone, I'm on behalf of Biocytogen. I would like to formally welcome everyone to today's webinar. We are very excited to introduce our pharmacology expert that will provide some insights into the capability and our technology platform here at Biocytogen so that we can ultimately support your ongoing research program. Before we continue, let me briefly introduce myself and the speaker for today. I'm Xiaoxuan and I'm from the marketing team at Biocytogen Beijing. I would like to honorably introduce today's speaker, Dr. Qianwen Feng, who graduated from Toyama University in Japan with a Ph.D. in life science and clinical medicine. She specialized in the in vitro screening of anti-tumor natural compounds and the mechanisms, especially the pathways of cell death and autophagy. In 2023, she joined Biocytogen as a pharmacology researcher, where she is currently responsible for in vivo and in vitro tumor pharmacology studies and the establishment of ADC-resistant cell lines. Participants may ask their questions in the Q&A box or drop us an email, and our representative would address the questions by email respectively. So now I would like to invite Dr. Qianwen to share her slides and begin her presentation. Thank you, and I hope you enjoy the presentation. Let's welcome Dr. Qianwen.
Hi everyone, my name is Qianwen Feng and I'm from the Pharmacology department in Biocytogen. Today I am going to introduce our ADC-related platform for preclinical research to fulfill your research need. And we will mainly share from these three parts, starting from ADC evaluation platform for IND application, and ADC and immune-oncology combination therapies, and lastly, BioMice models for ADC evaluation. Let me start with a quick introduction to antibody-drug conjugates, ADCs. These are targeted cancer treatments that use an antibody to deliver a powerful drug directly to cancer cells, while minimizing harm to healthy cells. The field is advancing fast. Several antibody-drug conjugates like Enhertu, Padcev, and Trodelvy are already approved, and many more are in clinical trials. With novel payloads, better conjugation techniques are strong momentum from industry leaders. ADCs are becoming a major force in oncology. As we often say now, everything can be conjugated. Firstly, we are going to introduce the ADC evaluation platform in Biocytogen, especially for IND application to start clinical trial. We have reviewed the studies that required for ADC IND submission. Here's the list of required in vitro experiments, and there are 22 of them in total. Items 1 to 5 mainly assess the antibodies affinity using methods like SPR or BLI, which we are currently not able to perform. Followed by items 6 to 20, which I am going to show at the next few slides, are the standard cell-based experiments covering cytotoxicity, flow cytometry for expression, and functional tests like ADCC, ADCP, and CDC. We suggest using the same cell lines as in the in vivo experiments for results consistently. And there are also some specific experiments like testing for the bystander effect, as well as MOA-related assays, including lysosomal colocalization, ligand blocking, signaling pathways, cell cycle studies, and apoptosis. And if it is a bispecific ADC, tumor selectivity assays are necessary to be performed. And item 21 and 22 are non-cell-based in vitro studies. Item 21 is the plasma stability assay, which evaluates payload release from the ADC over time in plasma for different species. Item 22 is the enzyme digestion experiments required for which is conducted by a third-party laboratory. The following section is the in vivo experiments required for IND application, which mainly include two types of cell line models, which are CDX and PDX. The PDX is derived from patient tumors, as well as the detection of PK/PD. This is the list of in vivo and in vitro experiments that can be performed by our Biocytogen platform, which involved a total 11 types of in vitro experiments and 5 types of in vivo experiments. Next, I will briefly introduce the detection methods included in the in vitro functional experiments. As for target validation, we use the antibody staining or absolute quantification beads to examine the target expression of cells by flow cytometry detection. We can also employ the IHC method to detect the expression in tumors. HER2 expression of 5 cell lines detected by flow cytometry. BT74 and NCIN87 have a high HER2 expression. RC40, this is RT48 at 1.11 microgram per milliliter, and to the 5 cell lines, and this is evaluation of the bonding of RC48 to at a series of concentrations, and calculated the EC50 of the 5 cell lines. As for ADC endocytosis, we use InQSight or flow cytometry to detect antibody internalization. InQSight is a real-time microscopy imaging system that supports both red and green fluorescence. The FibFluor is a pH-sensitive secondary antibody, labeling test article antibody, which releases red fluorescence when the antibody is internalized by the acidic environment of the lysosome. ADC cytotoxicity detection can also be performed using InQSight by analyzing the cell confluency to obtain the killing results. Detection methods such as LDH release assay and flow cytometry can be used to assess the functions of ADCC, ADCP, and CDC. Besides, we use confocal microscopy to examine the colocalization of antibody with lysosomes. We have a third-party partner to work for the confocal microscopy services. We can also detect the bonding and blocking of cytokines by antibodies using flow cytometry. The detection of proteins involved in certain signaling pathways can be performed using Western blot, such as PI3K, MAPK signaling pathways. Another unique effect of ADC is the bystander effect. After ADC kill the target cells, the payload will be released into surrounding environment and can kill the non-target cells. We can also co-incubate the target cells and the non-target cells with ADCs, and then detect the cytotoxic effects in both types of cells. Here is the the bystander effect of RC48 in BT474 and MDMB468 cell lines. Apoptosis can be detected using a Caspase-3-7 assay kit by InQSite. Also, niacin 5 PI staining to check the late apoptosis, early apoptosis, and the necrotic status. And PI staining can be used for cell cycle detection by flow cytometry. For the plasma stability assay, we incubate the ADC in plasma from different species, and then detect the release of the payload in the plasma. The plasma concentration of the small molecular will be measured by a third-party laboratory. And this is the third party for small molecular bioanalysis by LC-MAS. Next is the in vivo efficacy section. We have advanced CGenic platform featuring humanized mouse and cell models. In this example, the HER2 MC38 plus cell lines with humanized HER2 mouse model. We engineering a HER2 expressing CGenic system to evaluate the in vivo efficacy of HER2 targeted ADCs. Firstly, our humanized HER2 MC38 plus cell lines or tumor tissue have a positive HER2 expression. As shown here, both the DS8201 and trastuzumab MMAE demonstrated anti-tumor activity in this model, validating its utility for preclinical ADC assessment. With our extensive portfolio of humanized cell lines and mouse models, we offered powerful tools for translation oncology research. We currently have 152 CDX models, the cell line-derived xenograft models, and this number is still increasing. This is the list of our CDX models. The cell lines are implanted into our immunodeficient NDG mice. Here, A431 cell lines is a cell line with low expression of HER2, while BT474 and NCIH1975 CDX models are both cell lines with high expression of HER2. Our in-house DS8201 showed a very good efficacy in the BT474 and NCIH1975 CDX models, but it did not have strong efficacy in the A431 model with lower HER2 expression. We have also tested the efficacy of ADCs in PDX models, the patient-derived xenograft models, and the results have been very promising. And this is the workflow of PDX screening and in vivo study. Biocytogen has successfully established up to 270 PDX models on big NDG mice, which covers a total of 11 types of cancers. We also have experience working with autophagy tumor cell line models. On the right are the autophagy models that we are currently developing or have already completed. The in vivo bystander effect can also be detected. Here, NCI H1975 target positive cells and our Luke-Dodi cells, it is target negative cells, were mixed and subcutaneously implanted to the mice. The growth of target negative cells was detected by luciferase fluorescence imaging, and it can be observed that ADC2 in G3 group, G3 has a better bystander effect. The ADC PK detection includes two parts: antibody-related PK assays and small molecular-related PK assays. We have extensive experience in antibody-related PK detection, and we also have a reliable third-party partner to complete the small molecular PK detection. The second part is ADC and immuno-oncology combination therapies. For the combination therapies of ADCs and immuno-oncology, the IO, and we have two major models suitable for evaluation, which are syngeneic models and immune reconstituted models. The syngeneic models for ADC and IO combination therapies include wild-type mice with TA humanized cell lines, and both TA humanized mice and cells, or IO humanized mice with TA humanized cell lines. As for immune reconstitution models in combination therapies, both CDX or PDX models are applicable. Our humanized PD-1 mouse model can be used to test the efficacy of combination therapies involving immune checkpoint inhibitors. Here we presented the data on the combination of PD-1 and PD-L1 inhibitors with cisplantin. This model can also be used for ADC and IO combination study. We designed immune reconstituted models to test the efficacy of combination of PD-1 with ADC. The tumor cells used were NCIN87. It were inoculated at 3 days before PBMC treatment. And at day 0, we grouping the mice and injected with the PBMC, and then detect at the following days. And it can be observed that this combination in group 4 has a synergistic effect. The last part is a summary of the ADC-related models available from Biocytogen. We have over 2,000 TA target expression flow cytometry data, and RNA sequencing data, and in vivo tumor growth data from 269 human-derived cell lines and 23 marine cell lines. Additionally, we are also offering topic tumor models, spontaneous tumor models, metastasis tumor models, and 270 PDX patient-derived xenograft tumor models to fulfill your research need. And this is a list of tumor cell lines from human and C57BL6 background mice and BBC background and dogs. They are available in Biocytogen. And we also have reporter cell lines, which have been engineered to knock in luciferase. Here is the list of the cell lines with luciferase. The following slides present the list of some of our humanized TA cell lines, the tumor-associated antigen cell lines. And here is the list of some our humanized TA mice, which can combine with our humanized cell lines to make sure the expression of the target. Thank you for your interest in our service. If you have any questions or need further information, please feel free to contact us. And thank you once again for your valuable time. Have a nice day.
Thank you for your time and thank you for joining this webinar by Biocytogen. That's all for the presentation by Dr. Qianwen. If you have any question to us, please drop us an email at info@biocytogen.com and our representative will reply you shortly. Thank you once again and I hope to see you next time. Goodbye.